Okay, so now we're going to talk about the electron transport chain. So up to this point, we have undergone three main phases: the glycolysis, the preparatory reaction, and the citric acid cycle. And during those stages, we have formed a lot of coenzyme, that is NADH and FADH2, as well as made some substrate level ATP. What was the purpose of the first three phases? Well, it wasn't to make ATP, because we didn't make that much ATP. We only made a few molecules of ATP, right? But what we did make a lot of was these coenzyme molecules, NADH and FADH2. So the purpose of those three phases was to make those coenzymes, because they carry along with them electron and hydrogen ions, right? So in the electron transport chain, we're going to actually harvest those NADH and FADH2 so that we can grab onto the electron, and we're going to take those electrons and pass them from one carrier to another. And recall that electron has a lot of energy to it. So as we're passing them from one carrier to the next, we're going to release some energy, and that energy is going to be used to take those hydrogen ions that we also capture from NADH and FADH2 and form what we know as a proton gradient. What is the purpose of this proton gradient? Well, we'll see down the line that the purpose of that is to be able to create a lot of potential energy that we can then harness and convert that into ATP. One other thing that I want to mention is that we've been talking about how in order to even do these reactions and move onto electron transport chain, that we need to have oxygen, right? So if you recall, after in glycolysis, after glucose is converted to pyruvate, I mentioned that in order for us to move on into mitochondria, we need to have oxygen, versus if we don't have oxygen, we undergo fermentation. Why? Well, it's because oxygen is going to be the final electron acceptor. So as we take this electron and pass it from one carrier to the next carrier to the next carrier, that final carrier that we're going to pass it on to is oxygen. And when we finally pass it on to oxygen, oxygen is going to take that electron and it's going to accept it, and it's actually going to bind on with some hydrogen ion and make water. Okay, so let's kind of take a look at the electron transport chain into a little more detail. So the first detail is how are electrons delivered to the electron transport chain, right? Based on the name, you expect that electrons are involved. And we've already mentioned it, that the electrons are going to come from those coenzyme or electron carriers, right? So they're going to be coming from those coenzyme, NADH and FADH2. So as we pass these coenzyme molecules to electron transport chain, we're also passing these electrons. So that's how electrons are delivered. Why do we want these electrons? Well, we want these electrons because we're going to use that energy from an electron to be able to pump hydrogen ions. We're going to pump them from the matrix in the mitochondria into the intermembrane space. So we want to pump these electrons through. And by pumping, that means that we need some energy, because the protons don't want to move in that direction. They want to stay in the matrix. They do not want to move into the intermembrane space. So we're going to take the energy that we harness as we move the electron from one carrier to the next carrier. So you can imagine energy being passed, and then we take that energy and we pump some hydrogen ions through, and then that energy, that electron again gets passed to the next electron carrier, and we're going to pump again some more hydrogen ion. So this is going to form a proton concentration gradient, also known as the proton gradient. What is the purpose of forming this proton gradient? The purpose of forming this proton gradient is basically to be able to gather potential energy that we can then use later on by converting that potential energy into kinetic energy as we have these protons migrating back from the intermembrane space down into the matrix, and they're going to migrate through this molecule called the ATP synthase. So ATP synthase, just like its name suggests, is going to be able to synthesize ATP. So it's going to be able to add phosphate onto ADP as the hydrogen ions are moving through its space. So as the hydrogen ions moving, remember I said there's a lot of potential energy there. It's actually going to cause this ATP synthase to spin really fast. And so you can imagine as it's spinning, that's a lot of kinetic energy. So we can now convert that kinetic energy into combining ADP and phosphate to make ATP. So our goal with the electron transport chain is to make ATP. So let's kind of recap how it does that. So we have these coenzymes that has a lot of electron and hydrogen atoms connected with them. So NADH and FADH2 has electrons and hydrogen atoms that it can then pass on to the electron carrier. So it's going, NADH, for example, its electrons can be passed on to this first carrier. The carrier is going to take that electron and harness some of its energy and then pass it on to the next electron carrier. Because the electron, as it's being passed, has so much energy, some of that energy is used to push hydrogen ion from the matrix into intermembrane space. We're pushing it against its gradient using the energy from the electron to be able to push it against its gradient, resulting in a hydrogen concentration gradient. The hydrogen concentration gradient now has a lot of potential energy associated with it, so we'll say PE. And this potential energy is going to be used to spin the ATP synthase. So I'm just drawing it spinning here. And as it's spinning, and the hydrogen ion are being flown through or going through. So this is hydrogen wants to move in this direction, right? It wants to move from high concentration to low concentration. So as the hydrogen ion is being moved and is spinning this ATP synthase, some of the energy is going to be used to convert the ADP to ATP by adding this phosphate group to it. So all of this is happening right here in the mitochondria. So here's the mitochondria, and we can see that this is where we have the electrons being passed from one electron carrier to the other. And then you have the matrix, which is located inside right here. The intermembrane space is this space between the membrane right here. So it's kind of like right here and here. So again, we're pumping hydrogen ion as we're passing this electron from this space here to this space out here. But the protons don't want to stay in that space. They want to go back into the matrix. So they actually want to go back into the matrix. So as the protons are being are flown back into the intermembrane space, and into the matrix, and this happens right here with ATP synthase, that energy of it being flown through the ATP synthase will result in ADP forming ATP. So we end up being able to make a lot of ATP with the electron transport chain. So we can think about each time that you consume something, the purpose of consuming a carbohydrate is to make energy, right? That's the reason why we eat, is to make energy. And so we're going to convert, let's say that starch, that piece of bread that you eat, we're going to convert that into a bunch of glucose. Those glucose are then going to undergo glycolysis, preparatory reaction, citric acid, and electron transport chain. And the end goal of that is to be able to actually form this amount of ATP. So the maximum amount of ATP that we can actually form is 38. Typically, cells don't make the full 38 ATP. They'll make like 36 or less. They won't be able to make the full 38. And the rest of that energy that's inside the glucose, because glucose is a lot of energy. Just think about how each time you burn a marshmallow, when you go camping, how it just lights on fire like that. Lots of energy, right? So the reason why we do these series of steps is because if we were just to try to convert glucose straight to ATP, it would result in most of it being lost as heat. By taking these series of steps to convert that glucose into ATP, we're actually minimizing the amount of heat that would be lost, but we do lose a lot of heat. So a lot of that glucose is going to be, that energy right there that's inside glucose is going to be used to convert ATP, but a lot of it's also going to be lost as heat, right? We're just losing less heat by actually going through a series of reaction. So you can kind of imagine, a slow burn when you're roasting a marshmallow up high versus if you stuck it too close to the fire and we try to just harness as much energy as we can see the result of that. It's just that marshmallow just goes shoo, and we see this release of heat because of the amount of energy in it. So you can kind of imagine that if we try to convert the amount of food that we eat straight into ATP, a lot of it would be lost. Like we would probably be so hot, because we're just because so much energy is being released, that we would be probably so hot and probably less efficient too, because we wouldn't be able to make as much ATP. So we would probably be very slothy and tired and also really hot. That sounds miserable. Like I don't want to be like not have enough energy that I'm just sitting on my couch and at the same time being super hot, and just being like, oh my gosh, because so much energy is just being burst out from me trying to convert my food without going through a series of steps. We can also actually get energy from more than just glucose. We can actually also get energy from protein and from fat. That's also possible. The cell prefers carbohydrates though. It has a process through carbohydrate. It's much more efficient through carbohydrate. If we were to try to use either protein or fatty acid to make ATP, while it's possible, we would have to still convert it to the pyruvate product or the acetyl-CoA product. So you can see here that we could take the protein and we can convert it to pyruvate, or we can convert it to acetyl-CoA so that we can undergo these reactions. Same thing with fat, right? We can take those lipids that we consume and actually convert those fatty acids to acetyl-CoA, or pull off the glycerol off of the fatty acids and convert it up here in glycolysis to pyruvate. But it's just harder. It's not as efficient. So our body prefers that we start off using carbohydrate as our main energy source, and it's only when we don't have enough carbohydrate that our body would then be like, fine, I guess I can use protein or fat and oil, lipids. Okay, so that's the electron transfer chain, right? So let's kind of just really quickly recap. What is the purpose of electron transfer chain? Purpose of electron transfer chain is to take those coenzymes that we produced earlier in glycolysis, the preparatory reaction, and citric acid cycle. Take those coenzymes, harvest the electron energy and the hydrogen ions from it so that we can take the electron and go through a series reaction where we pass it from one carrier to another in order to produce that proton gradient so that we can then harness all of that potential energy into making ADP and phosphate into ATP. So in the end, we're able to produce a large amount of ATP per glucose molecule that we would then use to go for a jog, or to be able to digest our food, or be able to sit down on the couch and watch Netflix, right? So we constantly need ATP for our body to just be able to function properly. Where do we get this ATP? We get the ATP from actually the food item that we consume and converting that food item into ATP through a series of reactions. All right, so think about that the next time you eat this pizza. See you guys in the next lecture.